Okay, so we are starting Unit 4. And it was not like the best timing to start a unit, 2 weeks before finals, but I think I have a solution that will benefit us all. So, um, we are going to spend the next 2 weeks talking about cell communication and cell signaling and the cell cycle and mitosis. That's like what this unit's all about. Instead of a final, we're just going to do a unit test. So, probably that Monday or Tuesday, we'll do a unit test over cell cycle mitosis. I still want to give y'all some time to review the whole semester content, though, since all the semester content will be on the AP exam. So we'll probably use one of those regular school day schedules to take the test, and then spend the super, super long final schedule doing one big review activity over all of our units. Sound like a plan? All right, take some of that pressure off you for finals week, semester 1. So that's the plan then. We'll spend the next, this week is going to be all about cell communication and cell signaling. Next week is all going to be about cell cycle mitosis. So cell division, all that fun stuff. My goal for you for today is to describe what cell signaling is and the different types of external signals that cells can receive. Before I get started, does anybody have any questions? Apart from why is this room an igloo? But, all right. Cool, cool, cool. Yeah. Yes, there is always, just for everybody, there's always notebook paper in this first drawer right here. It's like a big stack of it, so help yourself whenever. There you go. All right. Also, this doesn't necessarily apply to you. Y'all are pretty good at getting your assignments in on time, but my last day for assignments turned in is December 10th. So, if you've got a missing exit ticket floating around out there, that's the day to get it in by. So, maybe my clicker will register. Haha. We've got our overarching idea for this lesson. Not necessarily the unit, and it's not necessarily our big ideas, like the AP biology big ideas, but this is if I were to sum up today in one sentence, this is what it'd be. This is what it would be. Cells can signal each other and interpret signals from both themselves and the environment. So, cells can signal and interpret signals from cells and the environment. And that's kind of what today is all about. How these signals work and where those signals come from. So, that's our big idea. We're going to talk about some examples. I think we're going to start off with an evolutionary connection because those evolutionary connections are super grand in the scope of AP biology. But this is our kind of big idea for today. But I don't like the word big idea because College Board has big ideas. Anyway, so cells can send signals and interpret signals from other cells and the environment. All right, so here is our evolutionary connection, and I know that is a kind of a beefy one. This is just some background information so you can understand the evolutionary connection. So I'm going to get my annotation thing up, and then I will highlight. There we go. So we're gonna spend this little example with yeast cells. So the way yeast cells identify mates is through cell signaling. Now, yeasts are single-celled eukaryotes. They can reproduce asexually, so just by themselves, but they can also reproduce sexually. There is a way for yeast cells to combine genetic information to create offspring. So, in order to do that, they need to find mates. So, yeasts are, single-celled organisms. They don't have the same kind of sexual dimorphism that humans have, but they do have a difference in two types. They are called alpha and A, which seems a little like grounds for confusion, but there's two types, alpha and A. That's really all you need to know. Each of them has a factor that one secretes and the other has receptors. So, right here, the alpha factor, the alpha cell releases alpha factors to receptors on the, thank you very much. Alpha cell releases alpha factors for receptors on the A cell. A cells release A factors that have receptors on the alpha cells. Yeah. This is what I do. If this is my A, that's my alpha. Yep. Getting those Greek letters down can be a tough one. So I just give it a little tail, and I make it a little more fancy. So this is kind of our background. This is an example of cell signaling, right? When an alpha cell is exposed to an A factor, and when an A cell is exposed to an alpha factor, what happens is they grow toward one another, and then they create a new cell that has a combination of their genetic material. We call these alpha A cells. Um, so this is how yeast cells reproduce sexually, and that is you only need what is highlighted because this is just background information for our evolutionary connection. Yes. No. That was just me emphasizing. We will be drawing a lot today, kind of a lot, but they're like relatively simple diagrams that I will draw out with you. So, yeast cells, mating types, background information. Now, here's where our evolutionary connection comes in. So, a species, biologically, and this is like all stuff, a species, biologically, is a group of similar individuals that can mate and produce fertile offspring. That's the biological definition of a species. So, if these mating factors ensure that mating happens within a species, then a change in the receptor or a change in the mating factor itself could lead to a new species, right? If a couple of these yeast cells have a mutation that changes that alpha A protein, that alpha A receptor, then those yeast would no longer be able to interbreed with the rest of the yeast population. So they would technically be a new species because they are able to reproduce fertile offspring between that small group, but not with the rest of their initial kind of parent population. So that's where we have the production of a new bacteria species, and therefore, like, another link in that evolutionary chain. So changes in cell signaling can lead to large-scale evolutionary changes. That's kind of our big idea. Questions here? All right. So here is kind of another evolutionary connection. So these signals, like once a signal reaches a receptor, we get something called a signal transduction pathway, which is a series of steps that happen after a signal is received. So, if a signal is like you hearing a doorbell, right? Ding dong. The series of steps that lead to a response, you opening the door, is you hearing it, getting up, walking to the door, opening the door. That's kind of like what a transduction pathway does to lead to a response. A lot of these pathways are very, very, very similar. We're going to talk about a couple of them, not tomorrow, but the next day. I guess tomorrow a little bit. So, because we have so many of these signal transduction pathways that are very, very similar, we think that these signal transduction pathways evolved in a very early common ancestor. Very much like the glycolysis evolutionary connection that we learned about last unit, right? How glycolysis is like the most overarching metabolic pathway. These signal transduction pathways are another piece of evidence for evolution. And we'll talk like an entire day on signal transduction pathways because they're really, really cool. Uh, but they're essentially that like kind of intermediate step between receive signal, response happens. So a lot of these pathways, they're very similar, specifically in like animal cells, but there are some very similar ones in like single-celled eukaryotes and like multicellular eukaryotes. So so we talked about like eukaryotes. I want to talk to you about prokaryotes for a second. This is a very common sensing signaling mechanism in bacteria, and it is called quorum sensing. So, what happens is that prokaryotes can actually sense the concentration of other molecules secreted by bacterial cells. So, one bacteria has like natural secreting signaling molecules. Other bacteria can sense how many other bacteria are around based on these signals, right? If every single one of us had a different strong smelling like perfume or cologne on, right? And one of us was blindfolded, they might be able to tell how many people are in the room by how many different smells there are around them. That's kind of like this. This allows our bacteria populations to coordinate. The example I have here is the production of a biofilm. A biofilm is a very, very thin layer. Well, to us, it's thin. To bacteria, it's think of like a multi-layered, like trying to avoid using the word film, but it's kind of like a you know how if you fall asleep without brushing your teeth, you wake up and your teeth have this like weird film over them. It's kind of gross. Yeah, that's a biofilm. It's created by bacteria and all of the sugars that are left in your mouth and all of the stuff bacteria have made living in your mouth. It's fine, perfectly normal. But it is a biofilm. It's a layer of sometimes mucousy, sometimes like mostly carbohydrate goop that where a bunch of bacteria live. They're really common in like ponds, where like that thin layer of like pond scum is considered a biofilm. And it's got a bunch of different bacterias in there working and living together. And the way they can coordinate like that is through quorum sensing. This is really cool. Questions here? All right. There are, now we're going to kind of pivot slightly or kind of focus in a little bit more on the different types of cell signaling, specifically external signaling. That's what we're doing today, is external signals. So there are three main categories of external signals that range from like super close contact to super far away. So there are contact signals, local signals, and long distance signals. And these are the three we're going to kind of specifically talk about. So, ranging anything from like cells literally being attached to one another to like your brain sending signals to like your toes, right? That's considered a long distance signal for like a cell. So, these are our three types of signals. If you don't get these down, they're going to be the titles in the next couple slides. So, here we go. The first one we've talked about before. and that is contact signals through cell junctions. So, if you remember, when we talked about how cells are connected to one another and how tissues are formed, we talked about cell junctions, those different ways that cells are like attached to one another. Cells can send signals through these cell junctions without crossing plasma membranes. So I recommend you kind of draw one of these too. We've got cell 1. This is going to be our signaling cell. And there's going to be a little cell junction. And it's going to send a signaling molecule to the target cell. The cell that is meant to receive this signal. This is a contact signal because those cells have to be, connected and touching one another in order to receive this signal. And it's through this cell junction, which is like a little connection between the cytoplasm. So these are really good in times of stress, right? When one cell is under stress, it is very easy to send that, hey, watch out, be prepared for this signal if it doesn't have to pass through plasma membranes. It also doesn't take any energy, like they can send these cells like via diffusion, right? If they like make this molecule that's in higher concentration over here, naturally it'll just kind of diffuse through the cytoplasm over here. So this doesn't take a lot of energy either. But this is a cell junction contact signaling. Questions here? All right, I've got another type of contact signaling, and that's just cell-to-cell recognition. Your immune system does this very, very well. So, in cell-to-cell recognition, two cells that are right next to one another, typically in an animal or a multicellular eukaryote, can communicate through interactions in molecules on their surfaces. So, for example, your in your blood types, that's what I'm trying to think of. Blood types, right? Your white blood cells, your immune system has receptors that recognize, okay, that A antigen, we know what that is. That's us if you're type A. They're like, okay, that's good. We're good. That is cell-to-cell recognition. Cell comes up to it, bumps into this cell, sees it's got the right antibodies, goes along its way. But if it bumps into something it does not recognize, right, that can be an alert to like, hey, this is bad. This does not belong here. Get this out. Destroy this. It could be very dangerous. So for this one, we've got a cell here, and it's got a little receptor thing. We've got a cell here, or that's a protein. This is our receptor. And we've got it right here. So this is going to be our signaling molecule because its signaling is on the outside. This is our signal right here. And this is our target. So these little doodles do not have to be complicated. But as long as you kind of get the meat of it, we're good. So this is our second type of contact signals. These are signals that can be sent when one cell physically touches another cell. These cells have to be touching for this to happen, right? Same with the cell junctions, right? They have to be physically attached to one another in order to make this happen. So those are our two types of contact signals. Cool so far? All right, we're going into local signaling. In local signaling, molecules are secreted by a signaling cell. So a signaling cell actually puts out the signaling molecules, like out into the environment, but they don't go very far. Um, these molecules are either meant to not go very far, they get taken up by all of the cells around it, or they're not strong enough to make it very far. So these are very, very local, uh, very, very short distances that these signals can be sent. And there are two types of local signaling, just as like there were two types of contact signaling. There are paracrine signals and synaptic signals. They're very similar, it's just where they happen. We've got paracrine signaling and synaptic signaling. Those are our two types of local signals. All right. Cool. So we're going to start with paracrine signaling. In paracrine signaling, a cell acts on a nearby target by releasing a local regulating molecule. Very, very similar to just like the general definition of local signaling. We've got a cell. We've got a signaling molecule. It's going to be released. It can be released, if it's too big, it will have to be released by exocytosis, which is like where we fold out the plasma membrane. Or it could, if it's small enough, it might be able to just diffuse through the membrane, or have a special channel. So we've got our signaling molecule. Or signaling cell. This is our molecule. This is our cell. And those little signaling molecules will cross a space into our target cell. Or on a receptor outside of the target cell. Sometimes the target cells like can't take in that signal because it's too big and they don't have the transports, but they do have external kind of sensors that can detect when they have that signal sent to them, and they can do what they need to do. So this is paracrine signaling. All right. So we got your signaling molecule sending off those little signals to their target cells. That's paracrine signaling. Synaptic signaling, I think, is super cool. So, in synaptic signaling, this happens specifically in nerve cells. And those signaling molecules are called neurotransmitters. So, these little signals right here, that's how your brain works. Also, how your nerves work. Also, how your muscles work. So, in synaptic signaling, a nerve cell releases a neurotransmitter molecule into a space called a synapse, which is just the kind of exit bit of our neuron. This will stimulate a target molecule. This is typically a muscle cell or another nerve cell. So if we're going to draw this, we've got our nerve cell or our signaling cell. And it has, there we go, this kind of end bit right here. They're called axons. And they can release these neurotransmitters. which will then be taken in by our target cell. So we've got our nerve cell. This is my friend, the nerve cell. This is the body of the cell. This is where the nucleus hangs out. We've got this long tail, and we've got these kind of frilly ends. What happens is it sends a signal all the way down this little tail here, and it spits the signal out down here. And then another cell has these kind of long growths on the body of the cell. Those are meant to receive those signals. And then it just kind of goes and goes and goes and goes and goes. Neurotransmitters are super cool. Usually just like, this involves a lot of like calcium ions and electrical impulses. But that's kind of out of the scope of this course. But it is kind of cool. So those are our local signals, right? Our paracrine and our synaptic signaling. Those are very short distances, but we do have to cross those cell membranes or worry about those extracellular spaces. So contact signaling, we didn't cross a membrane. Local signaling, we do, but we don't go very far. So we have one more type of cell signaling to talk about, and that is our like long-term signals. So those are our endocrine system. What do you think of when you think of the word endocrine? Anybody have any? Yeah? Muscles? Huh? Glands? That's a good one. Yeah. I think about hormones when I think about endocrine, because your endocrine system is what controls your hormones. But in reality, it's just like long distance signaling. This is the use of hormones for signaling across long distances in plants and animals. So, small organisms don't need this, right? They don't need long-term, like, head-to-toe signaling capabilities. So this is just in, like, plants and animals. And then what happens is we have specialized hormones or specialized cells that release hormones into the circulatory system or the bloodstream. And they travel to target cells in different types of the organism. So one of the more common and kind of universal experiences here is puberty, right? Puberty has a lot of interaction with that endocrine system, with releasing hormones, right? So most, if not all, of those hormones are made in gonad cells, right? Ovaries, testes, right? Those signals released there cause changes all over your body, right? Specifically, think like hair growth, right? So a signal that is made in like one localized place releases hormones into the body, and those hormones go everywhere and have different reactions on different types of cells. This is one of those. You also have hormones that regulate. They have a lot of effect on mood. They have a lot of effect on like heart rate and blood pressure. Your blood pressure is typically controlled by something that is similar to this. There's a lot of stuff that is controlled by endocrine systems. So if you were going to draw this, if you haven't already, you've got your endocrine cell, your signaling cell. It's got a nucleus. We've got our signaling molecule. We've got our bloodstream. Those signaling molecules enter the bloodstream, travel where they're going to go, and eventually reach the target cell. Woo. So that's our long distance or endocrine signals. Questions about endocrine signaling or any of our 3 external signaling types? All right. So, a little kind of sneak peek at what the rest of your week is going to look like. Today, we talked about the types of external signals. Tomorrow, we're going to talk about signal reception. So, these are our 3 stages of cell signaling, and we're going to hit all of these by the end of the week. So, the first stage is reception. So, that is the detection of a signaling molecule. We talked about why and how those signals are sent. We are going to kind of zoom in on just those receptors tomorrow, and we're going to talk about some of the specific receptors. Step 2 is transduction. We talked about that a little bit with signal transduction pathways. These are triggered by reception. So reception happens first, and then that kind of triggers transduction. What happens here is those signals are converted into a form that can bring about a response, right? Something inside the cell that says, hey, we got this signal. I think we should do this, right? That's our transduction. And then step 3 is response, what those signals actually bring about and how we can get ourselves to, do all this stuff. So, responses are triggered by transduction. So, this is a sequential series, right? We need to go in this order. We don't go transduction, reception, response. We don't go response, transduction, reception. It is reception, transduction, response. So, our responses are triggered by transduction, and there is a specific cellular response. There's one response that one signaling molecule tells that cell to do. So those we'll talk about how we can get those specific responses, how our cells know what to do based on which signals they receive. And this can be literally any cellular activity. From, hey, why don't you start mitosis, to, hey, you're not doing too hot, maybe we should just kind of digest you from the inside out. Or like, hey, can you send this signal farther down the line? Right? There's a lot of different cellular responses. Or, hey, maybe we pick up cell respiration, we need some more sugars. Or, hey, why don't you slow down? You're using too many resources. Literally, any cellular activity can be a response to some sort of signal. Questions here? Okay, so I went backwards. There's an exit ticket on Canvas. Be careful with the wording because I believe there are a couple compare and contrasts, which means you should have 2 main points in those answers. We have a little bit more time than you probably need, but that's okay. Just kind of settle into the week. If you have any questions, please let me know. This is day 1, and this stuff kind of builds. So, if you are lost, alone, vulnerable, confused, please hit me up. Let me know either now or via email or whenever tomorrow morning. I don't care. I am more than happy to either talk you through some stuff or give you some resources to help you move kind of through this week. Cool, cool.